Hello and a very warm welcome to everyone here in the LT and those joining us via the Zoom webinar. Thank you for joining us in this masterclass of pharmaceutical science. Now, before we begin, there are some housekeeping announcements. Please adhere to the safe management measures and maintain a safe distance of 1 meter apart. Sit in the designated seats here in the LT, which are the seats without a sticker behind them, and please keep your mask on throughout the masterclass. In the event of an emergency, please follow the directions of the ushers, which are myself and Anna in blue, and evacuate in an orderly manner. We also kindly remind everyone to please put your phone to silent mode. At the end of the masterclass, please exit the lecture theatre using the door on my right or your left at this end. And there will be a Q&A session at the end of the masterclass, but feel free to send in your questions anytime through the QR code or via the link polev.com Q&A LT26. That is polev.com Q&A LT26. For those joining us via the webinar, in the event that the Zoom session ends or disconnects, please rejoin us at the same link. This masterclass will delve into pharmacotherapy. You will gain an understanding of the simple concepts of pharmacotherapy and the application of fundamental biological knowledge in addressing real-life challenges. Our speaker today is Prof. Eric Chan, a pharmaceutical scientist at the Department of Pharmacy. Unfortunately, due to some unforeseen circumstances, Prof. Chan is unable to join us physically and will be conducting this masterclass via Zoom instead. Prof. Chan, let's give a warm welcome to Prof. Chan.
Thank you very much for the kind introduction. Yes, okay, thanks. A very good afternoon to all the students who are attending this masterclass at LT26, and of course those who are viewing the lecture remotely. Please accept my apology for not being physically at LT26, and the reason is because I've been tested positive of COVID-19 a couple of days ago. And now I'm recovering at home. But nevertheless, I'm still glad that I have this opportunity to share with you the masterclass via the Zoom platform. So one of the key objectives of the masterclass is really to demonstrate to prospective students who are interested to pursue pharmaceutical science in NUS an appreciation of how an interactive lecture is being conducted. And in our program, we conduct the teaching via various platforms, one of which is interactive lecture, which is what you're going to experience over the next 30 minutes. We also have collaborative learning workshop, where students will come together in groups to discuss a problem. And at the same time, we do have practical classes and also various research projects, internship that the students can embark upon. For today's masterclass, I would like to share with you a very interesting topic that is entitled Walking the Tightrope of Pharmacotherapy. And before I start, I thought I should briefly introduce myself. I'm now a professor in the department, and I'm trained as a pharmaceutical scientist. And pretty much you can see on this slide that my research interest pertains to pharmaceutical science research, whether it is in the area of understanding how a drugs work in the body, which is what we call pharmacology, or how a drug may cause adverse effect in the human body, which is the field of toxicology. And more intriguing is now there's an emerging understanding of how our gut bacteria may actually play a role in influencing the effect of drugs in the body. And I'm particularly interested in this interaction as well. In terms of how the body handles the drug, I'm particularly interested in how the body metabolizes the drug to form metabolites. And subsequently using knowledge in how our body handles the drug and how the drugs act on the body, namely termed as pharmacokinetics and pharmacodynamics. I'm interested then to use this knowledge in optimizing pharmacotherapy. So some of you may be wondering, what does pharmacotherapy refers to? The word pharmaco refers to drug, and the word therapy, of course, refers to treatment. Therefore, in terms of pharmaceutical science, we are constantly interested in how we use drug as a modality in treating diseases. Hence, I'm very passionate about how we can use all this knowledge in optimizing the pharmacotherapy of our patients and, of course, improving the outcome. Please also let me take this opportunity to introduce Dr. Yao Ping. She is with you at LT26 now. Maybe she can just stand up and wave her hand to the audience at LT26. So Dr. Yao, she is the Deputy Director of the NUS Pharmaceutical Science Program. And I'm very excited that she's actually physically with some of the students at LT26. Hence, after the masterclass, if some of you have additional inquiries related to our program, please feel free to interact with Dr. Yao. Like me, Dr. Yao is also an expert in pharmacokinetics, and she has a special interest in pharmacogenomic research. Again, pharmaco refers to drug, and genomics refers to the genetics of a human body. So she's interested to understand how genetic variability can lead to differences in drug response, and again, how that can be used to optimize pharmacotherapy. So this is actually a truncated version of an interactive lecture. And typically at the start, we will share with you the learning objective. And for today, the talk is entitled Walking the Tightrope of Pharmacotherapy. So in defining pharmacotherapy, one of the key factors that define how successful it can be is a field term as pharmacokinetics. Right? If pharmaco refers to drug, kinetics refers to movement, then pharmacokinetics refers to how the drug moves around in our body. And this is essentially defined by how the drug is first absorbed into our body, how the drug is carried in the blood to the different organs, how the drug is metabolized, say, in the liver, and how the drug is excreted in both the kidney and the liver, into the urine and the feces, respectively. And these processes, along with their kinetics, helps define how long the drug stays in the body, how extensive it enters the target organ, etc., etc. Hence, any unfavorable pharmacokinetic profile of drug can lead to unfavorable therapeutic outcome. And this include poor efficacy, which means the drug is not working at all, and we are not achieving our therapeutic target. Or worse still, we get to a state of potential toxicity, right? Where the drug now may be overexposed in the body and it reaches a level that becomes toxic to our biological system. So in order to illustrate this concept, I would like to actually use a case study of the first oral antiviral treatment for COVID-19, a drug with the brand name Paxlovid, to explain how enzymatic inhibition and induction may potentially modulate therapeutic and toxicity outcome. This is a short interactive lecture, but at the end, you should gain a very basic understanding of the concepts of pharmacotherapy and how the application of biological knowledge, such as how enzymes interact with drug or how drug interacts with drug target, can be applied to address real-life challenges. Along the way, I'll be using some quizzes to test your understanding, and I will encourage you strongly to participate in the quiz. The link for you to join the quiz is poev.com/ecyc. Maybe I'll take a slight pause down, and you may want to key this into your handphone. And you can use your name, or if you want to stay anonymous, you can use an abbreviation and enter this POEV. If you fail to capture this now, don't worry. Later on, my colleague can also type this into the chat so that you can look at this and participate in the quiz as well. When we talk about tightrope, what comes to our mind?
Hand in my hand, and we promise to never let go. We're walking a tightrope, high in the sky. We can see the whole world down below. We're walking a tightrope, never sure, never know how far we could fall. But it's all an adventure that comes with a breathtaking view. Walking a tightrope with you. Oh, 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 oh.
When you highline often, you really learn to train your fear like a muscle. Sometimes the height isn't the scariest part. Sometimes it's the exposure, which is how much space you can perceive around you.
Indeed, when we think about the tightrope, for some of us, the song Tightrope comes to our mind, right? I'm not sure how many of you has had the chance to watch The Greatest Showman, but it was a beautiful show and blessed with beautiful songs, right? I really hope I have the time to play the entire song so that you can enjoy it, but that is what comes to my mind when I think about the tightrope. On the other hand, for some of us, when we talk about the tightrope, we may be thinking literally about the tightrope that somebody walk, right, trying to cross from one part of a mountain to the other part of the mountain. Indeed, when we talk about the tightrope of pharmacotherapy, we are also talking about this very tight situation that we have to walk along, we have to tread along in order to ensure that the pharmacotherapy is successful. So before I talk about the tightrope, let me show you the graph on the left. You can see here, this is a graph where the drug concentration in the plasma of a drug is plotted against the time after dosing. And you can see that the drug actually increases as it's absorbed and then start to decrease as the drug gets distributed from the plasma into the organs and also get eliminated either in the liver or the kidney. And this plasma time profile of a drug, plasma concentration time profile of a drug is actually the outcome of the way the body handles the drug. And this movement is what we call as pharmacokinetics. So imagine if we continuously dose the patient over time based on a given regimen, we can actually start to see the accumulation of the drug concentration after every dose that's indicated as an arrow on the graph on the right. And you can see that given a regimen A, we will reach a steady state concentration where now this concentration falls within what we call a therapeutic window. The bottom line, the bottom dashed line here refers to the minimum effective concentration. And this is where the concentration of the drug needs to reach above and beyond so that we can get the therapeutic effect of the drug. However, if we now dose the drug using another regimen, say with an increased dose, right, by keeping the same dose frequency, right, we can now see that the concentration will reach an excessive adverse level, right? And this will lead to what we call a toxicity of the drug. Hence, the top dashed line here refers to what we call the minimum toxicity level. And this boundary between the minimum effective concentration and the minimum toxicity concentration is what we term as the tightrope of pharmacotherapy, or what we call the therapeutic window. Hence, if we dose the drug in such a way that the concentration does not reach this level, we end up with ineffective therapy. If it is excessive, we end up with toxicity. It's almost like a man falling either on the left or the right of the tightrope and ending up at the base of the valley. So this is actually not a good situation. Now let's switch gear and talk about the pharmacotherapy of COVID-19 infection. At the start of COVID-19 infection, many pharmaceutical companies and research labs, they were trying so hard to repurpose drugs for the treatment of COVID-19. And the reason is because time is an essence, and we really need to desperately find a cure. Hence, all previous antivirus drugs were tested either alone or as cocktail to treat COVID-19 infection. However, many of these drugs are not drug combinations. They are not successful. In fact, they can be potentially dangerous because of the toxicity. And as you can see here, I actually wrote an article in the Straits Times talking about how scientists all over the world were trying to repurpose the drugs for the treatment of this deadly infection. Fortunately, on 22nd December 2021, which is not too long ago, FDA actually authorized the first oral antiviral for the treatment of COVID-19. And HSA has also given an approval for the use of this drug. And we are waiting for the first shipment to come to Singapore. So very soon, COVID-19 patients who have the susceptibility to develop serious symptoms and also be hospitalized, they can be treated with this drug. So what is this drug? This drug is called Paxlovid. And you can see this is how the box looks like. And on the right, you can see this is how the tablets look like. So Paxlovid is what we term as the brand name of the drug. But the actual active ingredients contain two drugs, right? One is nematrelvir, which is stated on the box here as a codename PF073, so on and so on. PF refers to Pfizer because this drug is actually developed by Pfizer. And the second drug that is present in this Paxlovid is actually Rectovir. If you look at this packaging, you can see two beige or pinkish tablet and one white tablet. And this is actually a combination of two nematrelvir and one Rectovir tablet. So the question now is why? Why do we want to put two drugs into a single therapy that is used for treating COVID-19, right? So before I show you that question, keep that question in mind, and I want to tell you a little bit more about these two drugs. Nematrelvir is actually shown here, the chemical structure. It is what we call a 3-CL protease inhibitor. So the 3-CL protease is an enzyme that converts the coronavirus polypeptide into functional viral proteins. And this is a very important step that will generate viral proteins that promote the replication of the virus. Therefore, by inhibiting 3-CL protease, nematrelvir can stop the replication of the viral replication very effectively. In fact, if you look at the inhibition constant, it is 3.11 nanomolar, which is a very potent 3-CL protease inhibitor. On the other hand, Rectovir is a relatively established drug. It is actually a HIV protease inhibitor. HIV refers to human immunodeficiency virus. You can see here, this is the crystal structure of the HIV protease, and this Rectovir actually can bind and inhibit it potently. Similar to 3-CL protease, HIV protease is needed for the replication of the HIV virus. So now, I want to ask you a question. Please get ready your poll EV. So my question is, why are both nematrelvir and Rectovir included in Paxlovid for the treatment of COVID-19 infection? Yes, and I want you to participate as actively as possible. Do not worry about getting wrong, right? Because I have not even shared with you the answer. So this is really a teaser. Why? Why do you think nematrelvir and Rectovir are included in Paxlovid for the treatment of COVID-19? I'll just wait for maybe half more minute. The participation, by the way, is very active and very encouraging, and I'm very happy to see that. Yeah. Okay, maybe 5 more seconds. Okay, I will first review to you the responses, right? I will not tell you the answer, but I'll review to you the responses, all right? Okay, great. Yeah, I can see that the options are all chosen, right? All chosen, okay? And I will not discuss it now, but I will actually talk about it later, because these questions will come back to us again, right? This question will come back to us again. But I see quite a lot of you, 34% chosen that all the above are logical rationales, right? So if you have followed me faithfully until now, I do agree with you, right? All this could be logical, right? Rectovir has been found to be inhibitor of 3CL, possible. COVID-19 patients are more susceptible to opportunistic HIV infection, possible. Replication of COVID-19 virus is found to implicate HIV protease in addition to 3CL protease. Biologically, it is possible, right? Rectovir boosts the level of Rectovir in plasma. This one, I'm not too sure. No? Prof. Chan has not talked about it yet, but it may be possible. Okay, so this outcome is excellent, right? It creates the platform for us to discuss. So let me take a pause now and move on to tell you that drug is actually metabolized primarily in the liver of the human. And there are many enzymes that are involved in drug metabolism, but one class of enzyme is particularly important, and they are known as the cytochrome P450 enzyme, or in short, we call them the CYP enzyme. And there are different isoform and different family. So shown here in the middle of the chart, we have, for example, CYP3A, CYP1A2, CYP2E1, CYP2C, etc. Right. So these are all the enzymes that contribute to drug metabolism. If you look carefully, one of the enzymes that contribute quite a lot to metabolism of drug is CYP3A, right? And on the right, you can see that this is a crystal structure of cytochrome P450 3A4, or abbreviated as CYP3A4. Okay, so this CYP3A4 is very important because it's involved in metabolizing a lot of critically used drugs. So 3 key concepts. CYP3A4 is a hepatic enzyme. Hepatic refers to liver, that metabolizes drugs and also endogenous substances, right? CYP3A4 can be induced. What do I mean? Other drugs can actually bind to certain nuclear transcription factors, right? We'll not go down to the details, but other drugs can actually bind to those nuclear transcription factors, and then resulting in the liver cells making more 3A4, right? And in this case, the expression and the activity will increase. At the same time, 3A4 can be inhibited by other co-administered drug, right? And if it is inhibited, what happens? The activity will be decreased. Okay, the activity will decrease. So this slide shows you the structure of nematrevil in the middle and how it can be metabolized by liver enzymes to form various metabolites, namely M1, M2, M3, M4, and another dehydrogenated metabolite over here. The major metabolite is M4, highlighted here, and this metabolite also has been found to be having some 3CL protease inhibition activity. So that's quite nice actually, right? So over here, you can see that when they put nematrevil into CYP3A4 in increasing concentration, they can see an increasing velocity of the formation of M4, right? Which is this one here. Okay, they can see it forming. At the same time, when they measure this activity and they add an inhibitor of 3A4, which is known as ketoconazole, right? Which is the graph that I'm pointing to you now on the bottom left. You can see that the activity of this conversion of nematrevil to M4 decrease as we increase the concentration of this CYP3A4 enzyme inhibitor, which is a drug known as ketoconazole. And long story short is that they eventually found that the main enzyme that metabolizes nematrelvir is actually CYP3A4 in the liver, right? CYP3A4 is the key enzyme. And the other thing that they did is they actually gave the drug orally to human, and they found that the drug can be orally absorbed. So this is actually very good news, because you know why? So far, all the antiviral drugs that are approved to be used against COVID-19, they are all given by intravenous, right? Meaning they have to be injected, which is very, very invasive, right? But this drug, nematrelvir, can actually be given orally. So you can see here, remember this graph? We can plot the plasma concentration against time, and you can see the plasma versus time profile. Okay? But one of the big problems with nematrelvir is that the half-life is only 2 hours, meaning that within 2 hours, the concentration in the plasma dropped by 2-fold. Okay? Some of you may be wondering, is this a problem? From a pharmacokinetic perspective, this is a big problem, because if the half-life is so short, what it implies is that this drug has to be dosed frequently, every few hours, which is not feasible, right? You cannot be asking the patient to be taking Paxlovid every hour, right? Then it becomes a problem because compliance is an issue, right? So this is not ideal. So let me now introduce to you that ritonavir is actually a potent mechanism-based inactivator of 3A4. What do I mean by this? Okay, what I mean here is that essentially it is a very, very potent inhibitor of 3A4, right? The word mechanism-based inactivator here simply tell you that the way it inhibits 3A4 is so strong that it becomes irreversible, okay? The structure in the middle here, actually, the structure in green is actually the chemical structure of ritonavir, right? And the part that's highlighted red is the heme ion of 3A4. So CYP3A4 is actually an iron-containing enzyme, and this iron group or heme group is very important because the heme group is the one that is involved in the electron transfer, which is needed for all the enzymatic reactions. Therefore, when ritonavir binds irreversibly to the heme ion, it essentially kills the enzyme, right? It kills the enzyme. And what's the consequence? The consequence is the enzyme is dead, right? And you need to build more 3A4 in order to regain their metabolic functions, okay? Actually, ritonavir is not just a strong inhibitor of 3A4. It is also a strong inhibitor of other enzymes and also transporter in the body, right? So now, actually, I want to tell you why ritonavir is given along with nematrelvir. The reason is as follows. When nematrelvir is given alone, as I told you before, the half-life is very, very short. Therefore, we will likely end up to have low concentration of the drug and having ineffective therapy, unless we give the patient the drug every 2 hours or every 1 hour, right? Which is crazy, right? On the other hand, if we now give the nematrelvir together with ritonavir, can you imagine what happened? Ritonavir, remember, it is a mechanism-based inactivator of 3A4. It is going to inhibit 3A4, and consequently, 3A4 is not able to metabolize what? It's not able to metabolize nematrelvir, and therefore, the concentration of nematrelvir now becomes higher. And essentially, what we are trying to do here is to increase the biological half-life of nematrelvir when we dose together with ritonavir, right? Therefore, now we are able to push the drug level to be within the therapeutic window and therefore ensure that this is effective. Okay, so now I would like you to choose this again. Okay, this is the same question. This is the same question, right? But I want you to do this question one more time. Can you now try again and see whether what is the answer? Okay, can you please try again and tell me what is the correct answer? Okay, let me just look at the response now. Yes, guys, you should know the answer by now. Yes, absolutely. Right, what should be the correct answer, ladies and gentlemen? Yes, the correct answer is the one that is becoming highly selected by all of you, right? Which is Rectovir boosts the level of nirmatrelvir in plasma of COVID-19 patients. And that's the reason why we are making Paxlovid in this particular way, right? So now I want you to put on your thinking cap, right? Because I'm going to ask you two more applied questions, right? I'm going to ask you two applied questions, okay? Ladies and gentlemen, please get ready. Keep this concept in mind, and I want to ask you two applied questions, right? And this is especially important during our interactive lecture, right? Because we want you to learn a concept and then subsequently apply it in a new scenario. Okay, so are you ready? If you are ready now, I shall review to you the answer, right? So by the way, before I go on, I want to tell you, yes, the answer is this option. Ritonavir boosts the level of nirmatrelvir in plasma of COVID-19 patient. Okay, which of the following is a possible clinical scenario when Paxlovid is administered with another drug? Another drug that is a strong 3A4 inducer, right? There's a strong 3A4 inducer. Remember, 3A4 can be induced, right? And when 3A4 is induced, the level goes up, the activities goes up, right? So what could happen? Is it the plasma concentration of nirmatrelvir is reduced and the therapy of COVID-19 infection may fail? Or the plasma concentration of nirmatrelvir is increased and the toxicity associated with the nirmatrelvir may be observed in COVID-19 infected patient? Right, which is the correct answer? Is it the first one or the second one? Okay, again, I see a lot of active participation. Very, very good, right? Yeah, you guys are amazing, right? You are very, very responsive. Okay. I shall review the response. For the rest, you can still continue to choose. Oh, I have a split here. Okay, I have a split here. All right. So what should be the correct answer? Okay, when Paxlovid, right, is administered with another drug, say a patient has to take another drug for another condition, right, which is possible, and this drug is a strong CYP3A4 inducer. So what should be the situation? The situation is now CYP3A4 actually goes up, right? Right. Ritonavir is still doing its job to inhibit it, but CYP3A4 goes up now. So what happened? Given the same dose of Paxlovid, right? Now actually nirmatrelvir get metabolized faster, right? So what happened? The plasma concentration of nirmatrelvir is actually reduced, right? And COVID-19 infection may fail. So the correct answer should be this one. Okay, guys, are you clear? Okay, so this is possible, right? This is possible, and this is something that we'll think about when we treat our COVID-19 patient with Paxlovid. Okay, another challenging question. Okay, which of the following is possible clinical scenario when Paxlovid is administered with another drug X that is a key CYP3A4 substrate? So listen carefully. This drug that is given together with Paxlovid, right? It itself is a substrate, meaning that it itself is metabolized by 3A4. Okay, so now we are giving Paxlovid to the COVID-19 patient with another drug that is a CYP3A4 substrate. What will happen? Is it the first one, the plasma level of drug X, which is a CYP3A4 substrate, is increased and the toxicity associated with this drug may be observed? Or second one, the plasma level of drug X, which is a CYP3A4 substrate, is decreased and the drug is no longer effective? Which is the scenario? The first or the second? Okay, I'm very excited. Again, a lot of you are participating very actively. Yeah, this master class participants are really awesome, right? You guys are, I really hope that all of you can become our students because you are just so active in participating in the POEV. Okay, I shall review the response so that for those who have not keyed in, you can continue to key in, right? Yeah. Oh, again, I see a split. Oh, okay. Okay, you can continue to key in, right? But let me explain to you. So if now Paxlovid, which is our COVID-19 drug, is given with another drug X, okay, this is a circumstance, this is a scenario where the patient need to take drug X for some reason, right? But drug X is a substrate of CYP3A4. So what happened? Remember, Paxlovid contain what? Contain nirmatrelvir and ritonavir, right? And ritonavir is a very potent CYP3A4 inhibitor. So what happened? When ritonavir inhibits CYP3A4, right, it's going to what? It's going to affect the metabolism of drug X. Therefore, what happened? Therefore, the concentration, right, the plasma level of drug X, right, is going to increase, right? And this will lead to what? This will lead to, for example, toxicity of drug X, especially if drug X demonstrate concentration-dependent toxicity, right? So the correct answer will be yes, this one. All right. Okay, I'm really having fun. So in other words, I'd like to sort of summarize by telling you that indeed now Paxlovid is given to COVID-19 patient for about 5 days upon symptom onset. And the dosage is 300 milligram of nirmatrelvir. Remember that 2 pink tablets with 100 milligram of ritonavir. Remember that 1 white tablet. So every time we take 3 of that, right? And we will do this, what? We'll do this with all 3 tablets taken together. How many? Twice daily for 5 days. Okay? And look at this drug interaction. Co-administration of Paxlovid can alter the plasma concentration of other drugs. Remember, I told you about the inducer and drug X. So when we talk about walking the tightrope of pharmacotherapy, please keep in mind, we are not looking at one tightrope, we are looking at multiple tightrope. So as pharmaceutical scientists, we play a very critical role in defining all this knowledge, right? So that when a drug is given, all these tightrope can be carefully defined and the success of the therapy can be ensured. Where the patient is treated and the patient is not exposed to the toxicity. So I hope with this masterclass, you have learned the concepts of the tightrope of pharmacotherapy and how enzymatic inhibition and induction may modulate therapeutic and toxic outcome. So I would like to end by quoting myself, right? Walking the tightrope in pharmacotherapy is balancing between toxicity and lack of efficacy. As pharmaceutical scientists, we either ensure we have a very thick tightrope or we walk stably on a thin tightrope based on our innovations and knowledge. If you are inspired, join us in this important and exciting endeavor. Thank you very much for your attention. I'm happy to take questions now.
Once again, reminder to share in your questions at the poll EV link, which is Q&A LT26. Thank you.
I will also welcome students at LT26 to raise questions. You can actually use the poll EV. But if you have a question, maybe you can also put up your hand. Oh, okay. Wiping, do you have a mic? Do you want to handle this question? I'm not sure. Are you there, Wiping?
Hi, good afternoon, everyone. Yeah, so with regards to this question, what are the differences between pharm science and pharmacy? So for the pharmaceutical program, this is a 4 years direct entry honors program, right, under the College of Humanities and Sciences. And students would be learning more about the pharmaceutical sciences in terms of the understanding the foundations from the drug discovery all the way to the drug development process, and as well as the regulatory aspects, right, of the understanding the pharmaceutical industry. Okay, for students who are interested in the pharmacy program, that is also a 4 year direct entry program into the Department of Pharmacy, where you will be learning not only the pharmaceutical sciences, but also focusing a lot on the patient care areas, right, where you'll be learning about patient counseling, dispensing, pharmacotherapeutics, right, to prepare you as a pharmacist in the future. Right, so hope this answers your question, right, on the difference between pharmacy and the pharmaceutical science course. Thank you.
Okay, the next question pertains to whether the pharm science industry is becoming increasingly saturated. It really depends on whether you are setting aside for the Singapore market or you have a global mindset and you're looking at pharmaceutical industry globally. I'll say that the pharmaceutical industry would never become saturated if you are asking this question with the global mindset. In fact, if you look at the pandemic, many industries are affected, but one of the industry that thrives during the pandemic is actually the pharmaceutical industry, right? Many pharmaceutical industry are actually doing very well, and in fact, a lot more work needs to be done not only in the infectious disease spaces, but also oncology, neurology, aging, etc., etc. So I would very simply put it that the pharm science industry is not saturated, and I would also want to encourage the student who ask this question to maybe set your mindset beyond Singapore, right, and look at it regionally and globally. Thank you. I think the CHS program is a very wonderful program. I think the Vice Dean of Outreach or undergraduate at Dean's Office would probably answer this question by telling you that the program is flexible. In fact, many students have the options to declare one major, one minor, or even double major. And of course, the curricular structure is such that it gives you enough flexibility to select what you really like to pursue at the end of the day, right? So I would add that for pharm science program, you are not deprived of this flexibility. So I would say that come in with your mind open, eyes open. But of course, I would hope that you can choose the program carefully at the start. This class is very short, but after this, I hope you can continue to engage us either offline, or if you are at LT26 now, please stay back so that you can talk to Dr. Yao to verify if you are suitable for this program. Thank you. Our first inaugural batch is currently finishing their final semester. I do understand that quite a number of them have applied for a postgraduate program, such as MD program, say at NUS, and a couple of them have also applied for PhD programs as well. But I spoke to some others; they are more interested to start working in the industry. And there is also a small handful of them who are quite enterprising and are interested to start their own company. So I would say that it is not a case that most students will end up trying to do a PhD, but that is something that we hope some of you will be motivated to do eventually. Thank you. Oh, internship is very, very critical component of farm science program. So we have a lot of internship program. Maybe I'll take a pause because Dr. Yao is the manager of the internship. Wai Ping, you want to say a few words?
Sure. Right. So under the College of Humanities and Sciences, students are encouraged to take on internships under the undergraduate professional internship program called UPIP in short. Right. So this is not compulsory, but you're strongly encouraged to do so. Right. This would expose you to the pharmaceutical industry, get you prepared, right, for the work out there when you graduate. Right. So we do strongly encourage you to take on internships to better prepare you and to allow you to apply your knowledge that you learned in class to the actual application out there. Right. Thank you.
I would say that pharmaceutical science is both chemistry and biology, right? And I would say that mathematics as well, right? So when you talk about pharmacokinetics, we do deal with the math, and you cannot run away from statistic as well. So I don't have a magic number, but I'll say it's a good balance between chemistry and biology. Yeah, thanks. Just to let you know that I have 3 sons myself. My first son is in first year university now. My second son is in the second year army. So I exactly know how you feel, right? I can feel almost like you are my son as well. So truly, both of them actually forgot a lot of A-level stuff in the army, right? Somehow the army has this ability to clear away all the knowledge. But you know what? They come back very quickly as well. You'll be amazed how fast you can regain that knowledge after you join NUS, right? Or any other university. And I want to assure you that you can bounce back very fast. So don't worry about that. Yeah, we will help you. Don't worry.
Okay, that will be all for today's questions. And thank you guys for sending in such interesting questions and Prof. Chan for answering them. Should you have any other questions, do feel free to send an email to this the email here, which is askpharmscience@nusedu.sg. And once again, we'd like to thank you all for attending and paying attention. We hope you had a great time in this course and enjoyed a glimpse into your experience at NUS. Do feel free to explore more of Experience NUS Open House and the student life activities at UTown as well. We wish you a good day ahead, and please exit the lecture theatre by the door on the left. Thank you.
Thank you, everyone. Hope to see you soon in NUS. Bye-bye.